everybody. Welcome back to the second installment of today's lecture on the endocrine system. And we've discussed organs such as the ovaries as being responsible for producing hormones, progesterones and estrogens, your testes producing hormones in interstitial cells, some other organs that have hormonal type functions, the heart, the skin, the liver. We talked about vitamin D, the kidneys, the placenta. Also, the chemistry of hormones, they're not all the same. Hormones aren't all the same. Some of them are peptides, some of them are monoamines, some of them are steroids. And that influences how they function and are transported in the blood. So now I'd like to carry on with another aspect of the endocrine system, which is hormone receptors. So hormones are only as good as the receptors on their target cells. So hormones only stimulate cells that have receptors for them, just like the nervous system and neurotransmitters. Hormones stimulate only those nervous cells, or those target cells that have receptors for them. Say it's testosterone, cell, and the cell has a very specific kind of receptor for testosterone. And generally, that is the only thing that will be able to bind to that receptor is testosterone. Well, sometimes there are similarities. So that's how it turns out with the virus. The virus, it's similar enough that the virus can bind to that receptor, the ACE2 receptor, and then enter the cell that way. So there are similarities between different types of hormones and molecules. So what are the receptors made of? What do you think? What are receptors in a cell membrane made of? What do they consist of? Which kinds of molecules? Proteins, good, yes. And in particular, glycoproteins. Proteins? Yeah. And these glycoproteins or proteins will be in the plasma membrane. But not only the plasma membrane, also in the membrane, since we have an endomembrane system, and a lot of our organelles are enclosed or enveloped by membrane. So there are receptors within your cell for some of these hormones. For example, of the cell of mitochondria. Need to increase production of ATP? Welcome thyroid hormones. And other organelles as well. I also wanted to mention that, I'll put it in a little box here. Receptors exhibit specificity. Specificity. Say that 15 times when you've had a couple of gin and tonics. Specificity. Nope. I can't even do it, and I haven't even had any gin and tonics. Yeah, so they exhibit specificity, have very specific hormones for which they will bind. Also, there isn't just a couple. This looks like there's only a couple of receptors, but there are thousands. Thousands for a specific hormone. It's not just a one hormone, one receptor kind of system. And there is a phenomenon known as saturation. So say, for example, we have these 3 receptors here, and then we have testosterone in that receptor, testosterone in that receptor, testosterone in that receptor. Yeah, testosterone isn't the greatest example, but anyway, you get the idea. Now, all the receptors are saturated. So no more hormones can bind to any receptors because they already have hormones bound to them. So there is a saturation point. And there are some receptor defects. Diabetes type 2 is such that the receptor for insulin is deficient. So let's take a look at hormone modes of action. So we said before that if a hormone is a lipid, then it will have to be attached to a protein, a transport protein. But in order to be functional, it has to be unbound. So here's that same hormone unbound, but it's hydrophobic. It doesn't do well in blood plasma, but it just dekes right through the plasma membrane because the plasma membrane is also lipid and hydrophobic. So lipids and lipids, it doesn't even need to knock on the door. It just simply enters into the cell. As such, in this case, this hormone is binding to the nucleus, a receptor that's in the nucleus. That's a hydrophobic hormone. Yeah. Other hormones, such as this one here, a hydrophilic hormone. Well, it's great in the plasma. It's hydrophilic. It can just be dissolved in plasma, but it cannot get through the plasma membrane because the plasma membrane is lipid. So forget about it. It has to latch itself onto a receptor and from there instigate a second messenger activation. So a cascade of influences inside the cell itself. So hydrophobic hormones penetrate the plasma membrane, and hydrophilic hormones, the monoamines, cannot pass through the membrane. So that's why, I pulled testosterone off the top of my head for this example, but it's really not a very good example, is it? No, it's not a very good example. So let's cross out testosterone here because testosterone is actually a steroid, so it wouldn't have to bind to a receptor on the cell itself. So let's make that some monoamine. Sorry about that, my bad. So let's look at the thyroid in the system, the thyroid hormone. So there are 2 initially. The thyroid is hydrophobic. And the forms are T3 and T4. That refers to the number of iodine. There's drilling going on here. the number of iodines that this particular hormone has, 3 or 4. It turns out that T4 and T3 have slightly different processes. Yeah, so T3 binds, for example, to the mitochondria. It also binds to the rough endoplasmic reticulum. And why does that occur? Well, there can be various effects depending on what the cell is, where the cell is in the body. But it could conceivably just go right into the nucleus there and bind to a transcription factor in the nucleus and start the process of DNA transcription, thyroid hormones effects. So let me write some notes. Hydrophobic, steroid, and thyroid hormones easily penetrate the phospholipid bilayer on the target cell. And they just go right in and enter the cytoplasm. Enter the cytoplasm. Steroids enter the nucleus and bind to a receptor that's associated with DNA. This receptor has 3 functional regions. Three functional regions that explain action of the DNA. One region binds to the hormone. Let's see, no, that's not right. One binds to an acceptor site on the DNA molecule. And the other part of that receptor activates DNA transcription. So a receptor that has 3 modes of action for DNA transcription. And transcription produces new mRNA. And of course, that leads to the synthesis of a protein. Which then alters the metabolism of that cell. Yeah. So For example, here's a good example. Estrogen. Estrogen is a hormone that stimulates the uterine lining cells to synthesize receptors for progesterone. So it's very much a series of events that occur. Estrogen stimulates cells of the uterine lining, the lining of the uterus, to synthesize proteins that act as receptors to progesterone. They act as receptors. And progesterone, which comes later, will bind to those receptors it stimulates cells to produce glycogen. Long strands of glucose. And the glycogen prepares the uterus for implantation of an embryo. So it prepares the uterus for nourishment of the embryo. So that's one example. Hydrophilic hormones, because they cannot enter the cell through the plasma membrane, their mode of action is a second messenger system. So the hormone has to bind to a protein that's embedded in the cell membrane, and then there's a cascade of reactions. Inside the cell. So this is one type of second messenger system. And these are other types. So there's 3 different kinds of second messenger systems. They're not all exactly the same. So it depends on the hormone. So we said that there are many receptors for hormones. So there are many receptors for hormones. There might be a stimulus that causes the release of the hormone. The second messenger system will increase in volume inside the cell. So it's amplified. The effect of the hormones are amplified in the cell. And so now there's lots of proteins, enzymes that will activate other enzymes. So each time the effect is amplified. So in the end, you have quite a large effect. Yeah, it's called a reaction cascade. So it's quite a powerful system. How do you get rid of hormones? Hormone clearance. Eventually, you have to turn hormones off. They can't stay in the system indefinitely. So the liver and the kidneys, which are very good at this, the liver and kidneys take them up. They degrade some hormones. They're excreted. They're degraded and then excreted. in bile from the liver or urine from the kidneys. And hormones have a particular clearance rate. It's called the metabolic clearance rate, which is a very particular time period, and the half-life is the time required to clear 50% of the hormones. And that's usually what clearance is measured in. And hormones can be regulated another way. So not just the number of hormones, but the hormone receptors can be regulated. So if there is a low receptor density, there tends to be a weaker response. Hormone receptors can be upregulated. The number of receptors increased, and that leads to a stronger response. There can also be downregulation, where there's a strong response, but sometimes due to another hormone, then the receptor density is reduced, thereby reducing sensitivity of the cell and leading to a diminished response. Yeah, so for example, adipose cells. Downregulate when exposed to high concentrations of insulin and the testes cells downregulate with high concentrations of luteinizing hormone. Pharmaceutical doses can be somewhat problematic, like long-term use of pharmaceutical doses can cause target cells to convert to a different hormone. So, for example, if testosterone is overused as a synthetic hormone, the receptors can revert to receptors for estrogen, and that has feminizing effects. So athletes that use steroid hormones to increase their muscle mass, sometimes amongst other effects, one of them is, I think, depression as well. But because their receptors convert to receptors for estrogen, they begin to have a mammary glandular tissue forming breast tissue, for example. So hormones interact with each other. There may be synergistic effects working together. For example, FSH in males and testosterone are required for the production of sperm. Permissive effects, where one hormone enhances the response of another one. So estrogen, for example, stimulates progesterone receptors. In the uterus. And some are antagonistic, opposite effects. So those are, insulin is a good example of that, insulin and glucagon. Yeah, so insulin lowers blood glucose. And between meals, glucagon increases blood glucose, releasing it from the liver, for example. Yeah, so I'm going to end there. Thank you.